Hello everyone, welcome to Chapter 3 entitled Cells, the Living Units. It's a fun one for our anatomy and physiology class because it allows us to explore a lot of common biology themes that some of you may have already come across in your general biology courses. Now, for those of us who have never taken a general biology course before, no worries. This will be a really nice way to take a look at some of the main concepts that we encounter as we continue our journey into A and P. Now, the chapter is divided in various parts. For our first part, we're going to take a look at cells, primarily their plasma membrane. And we're also going to talk a little bit about the different forms of cell transport across that plasma membrane. So things like passive transport, active transport, osmosis, diffusion, tonicity. Now, before all that fun gets started, let's talk about the cell theory. The cell theory has three tenets or three parts. It basically says that based on everything we know about the workings of the cell and all the research conducted thus far, we know that a cell is considered the functioning unit of life. The second part is that all organisms have at least one cell. So here's the first part. The cell is the fundamental unit of life. And the second part is all organisms have at least one cell. So whether we're talking about our bacteria, which are unicellular, to our plants, fungi, animals, and our cells that are multicellular. The third tenet came about a little bit a little bit later, and part of that is because of the discovery of the microscope allowed scientists now to view items that we could not see with the naked eye. And that third tenet basically says that all cells come from other pre-existing cells. So that means that a cell doesn't spontaneously appear one day. There has to be another cell that gives birth or quote unquote gives rise to it. Now, before the discovery of the microscope and before this third tenet came along, many scientists believed, and now we're talking about the 1800s, about this thing called spontaneous generation. And spontaneous generation basically stated that if you had an inanimate object and you provided the right environmental conditions, spontaneously it would generate life. So, for instance, if you had some meat, if you just laid it out in the hot sun, it would spontaneously generate maggots. If you had puddles of water after a heavy rainstorm and you would just let them sit there, spontaneously it would generate mosquitoes. Frogs would spontaneously appear from puddles of mud, or areas that had high water, such as lakes, would in summertime spontaneously give rise to these little creatures that were hopping all around. Now that sounds very far-fetched and also very science fiction, right? But you have to keep in mind this was before we were able to zoom in and see that those frogs didn't just spontaneously appear. They were the end results of the sperm and the egg being released from frogs after external fertilization, and they are chemically attracted to each other in the water, allowing for fertilization to occur. And of course, that fertilized egg, the oocyte, will continue to develop until it starts to look something like a tadpole. And from the tadpole, it starts to metamorphosize into the adult shape. The thing you want to think about is when you take a look at the general shape of a cell, we're going to notice that it will always have a plasma membrane. Some of you might know this as a cell membrane, which is the same thing, and that provides the boundary or the distinguishments between the inside and the outside of the cell. Inside of the cell, you have what we like to call the cytoplasmic section or the cytoplasm. This is filled with a jelly-like fluid called cytosol, and within it you will find your different organelles floating around. If you bear with me for 1 second, I'll show you an image of a typical eukaryotic cell. And things like your nucleus, things like your mitochondria, things like your Golgi apparatus or complex, they are sitting within the cytosol flowing around the cytoplasmic region. And because humans have a eukaryotic cell, that would also mean that they will have a nucleus. And the nucleus, often people will say, well, that's the control center of the cell. And that is due to the fact that all our DNA, all of our chromosomes, are housed within the nucleus. And in a little bit, we'll talk about that anything that the cell needs to do, it needs to always go over to the genes or the particular sections of the DNA that have the information to make any type of secretion or functioning that it requires. So things like the production of enzymes, hormones. These all start with getting our proper piece of DNA so that we can follow those directions and produce our final product. Here is our typical eukaryotic cell. So let's start off with the fact that it has a beautiful plasma membrane that forms the boundary or the wall, separating it from extracellular versus intracellular. We see here in purple that we have our nucleus, and the DNA is marked by these darkened little purple section. It always looks like a little tie-dye event has occurred. And that, notice how it's been labeled as chromatin. Chromatin is a version of coiled up DNA. So it's sitting within that nucleus. And what else do we have? Oh, here in yellow, this is all cytosol. So that's the jelly that defines the liquid of the cytoplasm, which is this general area where you will find things like your mitochondria. Here's your smooth ER, rough ER, and the Golgi apparatus. So behold, a typical eukaryotic cell. As we kind of take a look at our cell, I want to talk a little bit more about our plasma membrane, because this barrier is going to allow us for the cell to become what we call selectively permeable or semi-permeable. And that has part to do with the fact that since the cell is the fundamental unit of life, it has nutritional demand, so we need to make sure that nutrients can get in, but it will also generate waste, so we need to make sure that it has a way to get those toxins out of the cell. So basically, we want to have a separation of what they like to call the intracellular fluid, what's happening inside, and the extracellular fluid, what's happening on the outside. The main component of our plasma membrane is going to be a lipid bilayer. And the reason they say it's a bilayer is because it's two layers that are stacked on top of each other. And this lipid bilayer is going to have what they like to refer to as a hydrophilic head and a hydrophobic tail section. And this is going to be very important because keep in mind our body, including our cells, inside and out, are mostly composed of water. So that means that you will have one aspect of the cell membrane, the head, that is hydrophilic or water loving, while the tail aspect will be water fearing or water repelling, aka hydrophobic. So let me show you what a typical shape looks like of this lipid bilayer. Some of you might know it a phospholipid bilayer because of its composition of phosphates and lipids. So here is what a typical membrane lipid looks like. And as you can see, it always looks, to me at least, like a little dancing head. You have the polar head, which is hydrophilic, water-loving, and then you have the tail that is going to be hydrophobic. Now, as I mentioned before, our membrane is a bilayer, so it's two of them stacked on each other. So it kind of looks like this. And this arrangement is very important because notice how the head sections that are hydrophilic are directly exposed either to the outside environment or to the inside environment. Why? Well, because that's mostly made out of water, and it can freely interact with the water in those particular elements. The tails, on the other hand, are folded inwards, facing each other, and that allows them to be shielded from the water or aquatic environment that the heads are exposed to. And that, of course, is because the tails are hydrophobic. This is what it looks like in a circular format. So once again, you can see that the hydrophilic heads are folded towards the outsides, whether it's the outside fluid or the intracellular fluid of the cytoplasm. And then the tails are tucked inside because they are hydrophobic. Now, this particular setup allows the cell membrane to then be considered semi-permeable, or some books will say selectively permeable. And what that means is that the cell membrane can control what comes in and comes out. If the membrane was fully hydrophilic, that would mean that everything could easily pass through. And if it was fully hydrophobic, that would mean that it would have a hard time accepting any type of movement through the membrane because of the fact of water that we expose ourselves to. The phospholipids are the main components, but they are definitely not the only components in a cell membrane. In fact, if we take a look at figure 3.3, we can see a beautiful illustration of the cell membrane. And here in light blue, I'm just going to highlight this particular section. You could see the hydrophilic heads of the phospholipid bilayer pointing outwards and the tails that are hydrophobic being tucked inside. But also what you might notice is that there are other things that are kind of inserted or embedded within the cell membrane. So here, for instance, these large purple structures, these are all different proteins that will find a home within the phospholipids and will serve a wide variety of different functions from the cell, anywhere from structure to communication, to response, to release or allow for transport into and out of the cell. In fact, if you bear with me on our next slide, I'm going to give you some examples of how these proteins can help us. You might also notice that it has these small little yellow structures embedded within it, and I went ahead and I highlighted it for you, that is cholesterol. Cholesterol is actually going to allow for the fluidity of the membrane to be altered. If you have just enough cholesterol, the membrane is perfectly able to control that semi-permeability. If you have too much cholesterol, believe it or not, the cholesterol molecules will actually start interacting with themselves more, and that will then leave the phospholipids to stiffen up, making it harder for transport to occur in and out of the cell. The other thing I want to tell you is that since our cell membrane is composed of primarily those phospholipids, but in addition also those cholesterols and other proteins, we like to refer to it as a mosaic model. Mosaic meaning it's made out of different pieces. And to be completely correct, they like to say it is a fluid mosaic model. And the fluid mosaic model, the fluid part, hints at the fact that the membrane is not stiff and can easily be manipulated in its overall shape, thereby maximizing its interaction ability with its environment. There are also some other fun things. Here I went ahead and I also highlighted what we call the glycocalyx, which are these like green, like little antenna things that you see sticking up from the cell membrane. Your glycocalyx actually plays a very important in your immune system because it allows your body to figure out which cells belong to you and which cells potentially can do harm, such as foreign cells like bacteria cells or virus-infected cells or cancer cells. Here is just a general look at some of the membrane proteins doing the different tasks as they are embedded within the cell membrane. This is just a little FYI as we proceed through the chapter. We're going to come across a lot of these again, and we'll talk about it in more detail. But for now, here are some examples so they can help with transport, opening and closing channels for ions to move through or solutes to move through. We can use it for signal transduction so that once the signal is received, you will have a conformational change within the protein, allowing the receptor to be activated, thereby allowing oftentimes a chain of different chemical reactions to occur and for the cell to become aware of the stimulus that it has received. We can use it for structural movement by utilizing it for part of the cytoskeleton, and this can be for the overall shape of the membrane as well as for scaffolding purposes so that you can move things like the mitochondria or in the case of cell reproduction, the chromatids, the sister chromatids, from one side to the next of the cell. It can assist with enzymatic activity, intercellular joining, which we're going to talk about in 1 second. It allows the cells to anchor towards each other. And of course, cell-to-cell recognition. And this is kind of what I was talking to you about, the little glycocalyxes, those little purple antennas that I was highlighting on the previous slide. Really important for your immune system to take a look at what cell is self versus non-self or foreign. Now, speaking a little bit about the cell junctions, when you take a look at the interaction of the plasma membranes of our cells, there obviously needs to be a mechanism that will hold the cells together and maybe even allow some type of interaction between them to occur. So go ahead and take a look at the illustration here. And what we see is that we have 3 cells that are sitting next to each other. And this makes sense because our body is made out of thousands upon thousands of cells. And what we see happening is as the cell membranes are next to each other, there are different type of attachment points that we can highlight. The first one is almost as if you are gluing or sewing the membranes together, creating an impermeable membrane. We like to call that a tight junction. And the tight junction, for lack of better explanation, literally means that you are super gluing two cells together so that they are joined together, but there is no other interchange happening within the cell. Now, what if you want to have a little bit of stretch? You want to have a little bit of flexibility? Well, in that case, we're going to look for what we call an anchoring junction or a desmosome. The desmosome is going to allow the cell, and I think this picture really shows it. It shows you how you have the cell membrane of the first cell. Here is that of the second cell. And then you have your connection points, your linkers. And what these proteins allow to do is to allow physical contact between the two cells, but it also allows both of the cells to stretch and pull slightly away from each other and then recoil or come back to their original position. It's fantastic for cells, for instance, like your muscle cells that need to be stretching a little bit so that they can return without being torn apart. Now, the third option that we have here is going to be called a gap junction. A gap junction is going to do two things. It is going to, and once again, I'm going to look at the picture as we're discussing this. Here is one cell membrane, here's the second. In a gap junction, it makes a physical connection point. So you're linking the two cells together, but by connecting them, you also do a second thing, which is that a channel will form between the cells that will allow cytoplasmic ions to move from one cell into the next cell. So gap junctions are excellent because they allow for communication through the use of different ions or solutes within the cytoplasmic content. So for instance, if we're going back to our little muscle cells, these muscles, in order to contract and relax, require lots of sodium, potassium, calcium. Well, if they have a gap junction, which many of them do, they will be able to share these ions from one cell to the next. It is quite common for cell-to-cell contact to involve a multitude of different junctions. So that's why when you look at the illustration on top, you might often notice that you'll have some desmosomes or anchoring junctions, as well as some gap junctions, a little bit of tight junctions, a little sprinkle of everything all over the place. Anything that can be utilized to connect these cells, allowing for a physical and sometimes a chemical form of communication between the cells. So now that we are a little bit more familiar with the plasma membrane and we've talked about the fact that it's selectively permeable, let's talk about how exactly do we get substances, so little ions, little particles, little solutes, water, how do we get it to move across the plasma membrane? And it seems like we have two general options to look at. We have what we call passive process and we have active process. And many of this is based on a concentration gradient. A concentration gradient highlights the fact that your cell membrane creates a border between the outside and the inside of a cell, creating two different environments, internal versus external. If we were to look at an ion or a solute, if we notice that they have a concentration gradient, that would mean that their quantity or their concentration would be different internally versus externally. So for instance, on the illustration that is right over here, we go ahead and just erase these little marks right here. On the illustration that we have at the bottom of the page, this particular black line highlighted in yellow, and with me now with my red over, that is going to represent my plasma membrane. And we can go ahead and assume that this is the inside of the cell, and this will be the outside of the cell. The little red dots will be used as an example of a solute that has a concentration gradient. It has a difference with how much it has inside the cell versus outside of the cell. Now, I hope that you'll agree with me that outside of the cell, there are a lot more solutes than there are on the inside of the cell. And what we see happening is that if the solutes fit through the plasma membrane and they start moving down the concentration gradient, so from high to low, this would be an example of passive transport, because passive transport allows for us to go down the concentration gradient. It does not require any ATP, so ATP is not required when we do passive transport. Instead, its driving force is going to be kinetic energy. Kinetic energy is the energy of motion. The cell does not have to do any additional work to have access to it. Within your passive transport, you can go ahead and narrow it based on, are we moving solutes or particles, or are we moving water? You might notice on your notes that on their passive processes, it's been highlighted that you have what we call simple diffusion, and we have facilitated diffusion. Both of these mean that you are moving around solutes or particles, and they're going down the concentration gradient. So if you have a diffuser in your home, that usually entails that you have some type of container where you've added some fragrance, maybe an oil form to it, and it will either spray consistency or it will diffuse into the environment. So maybe you have like little bamboo sticks that you insert in it, and the liquids will start interacting with the bamboo, allowing the scent molecules to go from high, the origin point, your little storage mechanism of these liquids, to low, which is the environment that it's in. Even if you place some perfume or some cologne on you today, the reason you did that is because you want to smell nice. But you also want people to smell how nice you smell as you walk by. And that's possible because the scent molecules diffuse. They go from an area of high concentration, because you were where the scents were deposited, into low, which is the surrounding environment. The difference between simple and facilitated diffusion: simple diffusion means that the item can fit through the cell membrane without any type of assistant. This will usually be a lipid-soluble item so that it can gain quick access to the inside of the cell or the outside. It depends on how it's moving down its concentration gradient, right? Osmosis, facilitated diffusion means that there's usually a channel or a pathway or a carrier that has to be utilized. So once again, the solute or the particle in facilitated diffusion will still move from a high to a low concentration, but it doesn't naturally fit through the plasma membrane, so it will need to utilize an additional protein or carrier for it to make it through. And hold on the next slide, I'll have some illustrated examples for you. Diffusion, whether it's simple or facilitated, will always be involving the movement of solutes or particles. And if you're interested in moving water, we would then use the terminology osmosis. Osmosis is the movement of water from high to low. So passive transport, we're going down the concentration gradient. We're going to be using kinetic energy, and we're going to refer to it as diffusion or osmosis, depending on if we are moving solutes or particles. Solutes or water, solvent. Now, active transport is going to involve the movement from low to high. So you're going up the concentration gradient, you're going against the concentration flow. This usually entails solutes that cannot move by themselves or fit by themselves to the membrane. And in order to make sure that everything gets done, active transport will require ATP. Kinetic energy is not enough to get the work done. ATP required. So here, for instance, we have an example where, once again, here's our cell membrane. And you can see in the first two, we have an example of simple diffusion and carrier-mediated facilitated diffusion. Oh, I forgot about the third one. Channel-mediated facilitated diffusion. Because you have the term diffusion in all three, that means that you are moving a solute, a little particle, and if that solute is lipid-soluble and it can fit by itself, it will move from a high to a low concentration utilizing kinetic energy, not ATP. If we see that the solute has to move through a protein, and it has to, in essence, fit within the protein carrier before it can make that journey, we call that a carrier-mediated facilitated diffusion. Once again, not requiring any ATP, simply requiring kinetic energy. And then our third option is the channel-mediated diffusion. So now, instead of having a carrier-mediated where the solute has to personally interact with it to open and close, there is a protein that simply provides a channel. Notice on the illustration how the channel is continuously open, and that is because it is waiting for the flow to go from high to low. So those ions can diffuse down their concentration gradient. The fourth option, you might notice water molecules. So we have water, and the water is moving from high to low. So that would be an example of passive transport as well, but it would also be a better example of osmosis, the movement of water from high to low. There are some beautiful illustrations as well as tables in your book, and I definitely recommend that you check them out. So for instance, here it's giving you a nice comparison between simple diffusion, movement by itself, facilitated diffusion, which is going to be using either carriers or proteins or channels, and then osmosis, which will solely be the use of or the movement of water. Notice that all three examples will utilize kinetic energy. No additional energy is required. Now, our body is indeed mostly made out of water, but there are some other solutes floating around, of which sodium is actually the most common. So when we look at the concept of osmolarity, what we're taking a look is what is the total concentration of solute particles that we expose our body, but more importantly, our cells to. And the thought process behind that is because the solution is a mix of a solute, a particle, and a solvent, which is water. And the relationship between the solute and the water is inversely related. When one goes up, the other one goes down, and vice versa. Let me give you a very simple example. Let's assume that we have two beakers that are filled with a solution of sugar and water. If one beaker has 40% sugar, that would then automatically mean that it has 60% water. Now, what happens if I say, you know what, my sugar, instead of 40%, I want to make it 80%. If something is 80% sugar, that would then mean that it's only 20% water. And we see this relationship and osmolarity go along all the time. If one is high, the other one will be low. Most of the time, they won't tell you both of them. You are just assumed to know that it's an inverse relationship. So if you're doing your homework or you're reading up on it and you see the mention of what they call a high concentration of solutes or a highly concentrated solution, then the fact that you know that it has high solutes, that means that it will have low solvent or water. And if it says it is a low concentration of solutes, then you would know that it has a high concentration of solvent or water. The reason you want to keep this in mind is because water will go from high to low. So here it says right here on your slide, we switch back to my red. It says water moves by osmosis from an area of low solute to an area of high solute. Because if the solute is low, the water is high. And if the solute is high, the water is low. So water will always go from high to low. So when you do your tonicity and you're comparing how different environments can affect the cell, you want to keep in mind the relationship between the solute and the solvent water is inversely related, and water will always go from a high to a low point. This, of course, is important because some solutes will not be able to pass through the plasma membrane because of a wide variety of reasons. They might be too large, they are going against their concentration gradient, anything can happen. So you want to keep in mind that anytime you have a difference in movement, it can cause a shift or movement of water, either mostly into the cell or out of the cell. Let me show you this example. So here we have an illustration where we have a tube, a U-shaped tube set up. Here in yellow, we have our permeable membrane. And the reason that they wrote freely permeable is because the openings are so large that the water and the solute, these little red particles, can easily move across. So both of them fit. So that means that if we look at our left side, it says the solution has a lower osmolarity, which is indicated by less little red circles, which means that it has a high amount of water, because if one is low, the other one is high. And then on the right-hand side, it says you have a solution with greater osmolarity. Once again, we're looking at our little red dots. Notice how these vastly outnumber those on the other side. And if it has high amount of solutes, that means that it has low amount of water. Now keep in mind, water loves to go from a high point to a low point. Notice how the movement in the black arrow is indicating that high to low movement. And as the water is moving, the solutes will move as well from high to low. And since both of them fit, the movement will continue until we see that we have both solutions at the same osmolarity. So the same concentration of water on both ends, the same concentration of solutes on either end. This is then a perfect example of equilibrium, where the volume with the naked eye is indicative that both the particles and the water are equal. So keep this in mind, and then let's take a look at the example. In this particular example, you have a selectively permeable membrane, and once again, you have two separate environments. You have your low solute to the left, and you have your high solute to the right. Now, because the membrane is selectively permeable, that means that the solute cannot pass through it. So water can easily move through, but the solute cannot. So the red dots are going to stay where they are, but the water will move accordingly. What's going to happen? Well, if you have low solutes, then what we were just chit-chatting about, that means you have the opposite. That means you have high water. And if you have high amount of solutes, that means that on this side on the right, you have low water. So if you put water in a situation where it has a selectively permeable membrane and it has a concentration gradient, an area where it's high and an area where it's low, we see that if the water can fit, it's going to go from high to low. So over time, the water is going to move from the left to the right. And what we'll see happening is, yes, the volume will get pushed upwards and the water will continue to move so up until the point that it has identical osmolarities, so identical ratio of water to solutes. But in order to do that, the water will have to move with a higher rate over to where it was initially at a low concentration until it's at an equal concentration based on the osmolarity of both the solutes and the water. So keep in mind that even though the volume of this illustration, you're looking at it going, the right side has more than the left side. Yes, but the osmolarity, the concentration of solutes and solvents, particles and water is equal on both ends. How does this then apply to our cells? Well, keep in mind that part of our homeostasis is that our cells are continuously being exposed to different type of solutes or ions or concentrations. So part of what we call tonicity is to take a look at how water can move across the cell membrane. And given different circumstances, you can have a mass movement of water into the cell or out of the cell, and the cell shape will be affected accordingly depending on how it will move. So we have three scenarios. you have what they call an isotonic solution, a hypertonic solution, or a hypotonic solution. Iso means the same. In an isotonic solution, what we see is that the osmolarity is the same on the inside of the cell as it is on the outside of the cell. Best example of that is physiological saline. When a patient goes to urgent care or it goes to the hospital, oftentimes they are given quote unquote fluids. Fluids are usually water based, but a very tiny amount, like 0.9% sodium chloride, just a tiny little bit of salt, which is perfect. This is very similar to the fluids that we naturally have within our body, so we like to say this is isotonic. If the osmolarity of the solute is the same inside and outside, then the amount of water will be the same inside and outside. So what we see happening in an isotonic solution is that the volume will remain unchanged. Will you have water move? Yes, because water is always moving. That's that kinetic energy. But whatever goes in comes right back out. One thing that I want to emphasize is please keep in mind that when we discuss these isotonic, hypertonic, hypotonic solution, the water is the only one moving. The solutes stay within their original environment. They do not fit through the selectively permeable membrane. So isotonic, everything is the same. Whatever goes in comes right back out. And this is when the cell is usually in its natural shape or form. Hypertonic is when you have a solution that has a higher amount of osmolarity or solutes on the outside of the cell than you would encounter inside the cell. So remember our little physiological saline, which was like 0.9 sodium chloride? Well, what if we take a look at a 10% sodium chloride solution, a high salt solution? Well, our body is not composed of 10% sodium chloride. So if our cells are exposed to these cells, this environment of 10%, what we see is we have a hypertonic solution. And if there is more solutes, that means that there is less solvent. And this will then cause the water to be drained out of our cells. In a hypertonic solution, water comes out of the cell, allowing the cell to what we call crenate or shrink in its overall size. So isotonic, we stay the same. Hypertonic, the cells become smaller because water comes out, because the solute concentration is higher in the solution than it is inside the cell. And our third example will be hypotonic. Hypotonic is when you have a low amount of solute on the outside of the cell, meaning that you have a higher concentration of water on the outside of the cell than you have inside the cell. So for instance, pure DI water, which is just nothing but H2O molecules, if we expose our body to that, we are not made out of 100% water. So that water has nowhere else to go but into the cell. So in a hypotonic solution, water goes into the cell because the solution has less solutes and more water. And as the water goes flooding in, the cell starts to swell up like a balloon being inflated. And because we have a cell membrane, it could eventually lyse or pop open. Scenario looks like when we're looking at it under the microscope. Here's my typical red blood cell. Notice how it has its little biconcave shape. If it's placed in an isotonic solution, then everything that is the same on the inside is the same on the outside, and vice versa. So notice these little black arrows. Whatever water moves in, moves right back out. So we have a normal-sized shaped cell. In our second scenario, we have a hypertonic solution, and that means that we're exposing ourselves to a solution that has a high amount of solutes on the outside and less water on the outside than it has on the inside. So water will always go from high to low, which means that the water will leave the cell. And notice, once again, the water is the only one that's moving. and the cell starts to deflate and it starts to get these little sharp corners, we call that crenation. So anytime you hear the word crenation, that means that water is leaving the cell, it's dehydrating the cell, and that could be because it's been exposed to a hypertonic solution. Last but not least is a hypotonic solution. Hypotonic solution means that you have less solutes on the outside, which means that you have more water on the outside than you have on the inside of the cell. So that water has nowhere else to go but into the cell. And notice how my little red blood cell starts to swell up. And if it keeps going, it will eventually lyse open. We call that hemolysis. Hemo for blood, lysis to pop open. Those were all examples of your tonicity, of your osmosis, your diffusion. Those were all examples of passive transport, where your kinetic energy was simply allowing for the movement down the concentration gradient or from high to low. I would now like to go ahead and switch over to active membrane transport. Now, keep in mind, when you're doing your active transport, that means that you are going against the concentration gradient. So you're going from a low to a high concentration, and you will always require ATP to get this done. Some of the examples that we'll talk about will be the sodium-potassium pump, which your cells utilize all over your body, and they require a massive amount of ATP to maintain consistent as part of homeostasis. And we're also going to talk about vesicular transport, which is the building of a little vesicle to move items into and out of the cell. Here are some examples of how active transport will come into play. Usually it's needed because the solute, the particle, is too large. It is not lipid soluble, and of course it's moving against the concentration gradient. It is going from low to high. And please always remember that you are going to require ATP to get the movement going. We can take a look at different carrier proteins as examples of active transport. These carrier proteins will often move more than one substance, and you'll see the terminology of an antiporter versus a symporter consistently throughout the chapter. An antiporter means that one substance will move in while the other moves out. So you'll have two opposite directions moving in. In fact, your sodium-potassium pump, which we're going to see very shortly, is a beautiful example of an antiporter. Symporter means that these two items can move simultaneously in the same direction. So this is like they're either both entering the cell or they're both exiting the cell. Once again, it's still going against the concentration gradient. It's still requiring ATP. Here is our sodium-potassium pump that I was mentioning to you. So I want to start off by giving you a little clear orientation of where we are. This is the cell membrane. We can see the hydrophilic heads and hydrophobic tails. We have our pump, our little protein embedded. And what's going to happen is with the use of ATP, here we go, there we see our little ATP molecule, we're going to utilize active transport to continuously kick 3 sodiums out and allow 2 potassiums back in. So as soon as you see ATP, you know that this will be active transport. In the case of this, you can also say it's an antiporter because the sodium is moving outwards and the potassium is moving inward. Here are some other examples of how ATP can be utilized as a mechanism of movement. In this case, this is what we like to call a secondary active transport. And it's going to be used to create a secondary concentration gradient that will allow a molecule to kind of piggyback and move along the way. So starting on the left-hand side, you're going to see that you have your protein pump and utilizing ATP, which just makes it part of active transport. We're going to see that we have a movement of sodium going into the cell. And what's going to end up happening now is as the sodium is being pumped out of the cell, it's going to create a concentration gradient that will allow for the glucose molecule to piggyback and follow it as it naturally moves into the cell. Now, because the glucose moved together with the sodium, that would then be an example of a symporter. They're both moving in the same direction. And I know some of you might be looking at the illustration going, but there's no ATP. So how can this part be active transport? You're correct. This particular section of the transport doesn't require ATP, but this part wouldn't happen unless the first part happened. And since the first part required ATP, that means that the second part is enjoying the benefits of utilizing that energy, which would then still mean that it would be qualified or classified under the active transport, but we call it secondary active transport. Another example of where we could come across the use of our ATP molecules is going to be for vesicular transport. This is when we use pieces of the cell membrane to create a little vesicle that can be used to move items into the cell. We can also go outside. So we have what we call endocytosis versus exocytosis. Endo is the movement of the cell, movement of the solutes or liquids into the cell, and they're being transported by the little vesicles. Exocytosis is when the item is being exported out of the cell. Both of these will require ATP. Sorry about that, ATP. Now, let's take a closer look at endocytosis because you have different classifications, subdivisions before that. The first one is what we call phagocytosis. Phagocytosis is the movement of large entities into the cell. Oftentimes, it will be referred to as bulk eating. If you take a look at the illustration on top, you're going to notice that this is the cell membrane. And whenever there's an item, for instance, right here, that we're trying to move in, what we see happening is that the cell membrane will fold inwards, encapsulating its object of choice that it's trying to introduce into the cell, and it will pinch off a piece of the membrane, forming a little vesicle. If the item on the inside is a larger solid item, we can say it is it's doing phagocytosis. If instead of a large item, you have a very minute, a very thin solid, or better yet, a liquid, then what we say is it's penocytosis. Penocytosis is also known as bulk drinking. Once again, you'll still need to form the vesicle, but now on the inside of the vesicle, you usually have liquids or very minute solutes that are moving in. The third example of endocytosis is what we call receptor-mediated endocytosis. And as the name indicates, there are some receptors that need to be activated before we can form the vesicle. So here in my illustration, I have my little green solutes that need to interact with receptors that are embedded within the cell membrane. If they fit and or activate the receptors, the vesicle will form and will engulf them to allow them to successfully enter into the cell or cytoplasmic area of the cell. A really good example of a virus that utilizes this model is the HIV virus. The HIV virus interacts with receptors found on T helper cells, and once the interaction is made, the virus is then engulfed into the helper cell, and that is then how it gains access to your genomic material, meaning that it can use your DNA to start replicating and increasing its viral load. Researchers are currently trying to develop a vaccine that would hide these receptors from the HIV virus. And if it hides them, that would then mean that the virus wouldn't be able to interact with them. And if they can't interact with the receptor, they can't induce endocytosis. And because it is a virus, it needs a host cell. So if it cannot gain access to the host cell, that virus will perish and it will not harm the body. Would be truly an effective way to eradicate HIV. And our last slide for this particular part is going to concentrate on exocytosis. So endocytosis is when we form our vesicles and we enter into the cell. Exocytosis is when we have a vesicle that now needs to exit the cell. Once again, we have items that we are transporting. We can use this in the case, for instance, whenever we do enzymes or milk that has to be dumped into the milk ducts, hormones that have to be released into the bloodstream. And what we see happening with exocytosis is that the vesicle will go towards the plasma membrane and will interact with the snare proteins, which will allow them to anchor. Once they're anchored and fused, we see that the vesicle will form a small opening, allowing the excretions to be released into the extracellular plasma, into the extracellular fluid, and the vesicle will now become part of the plasma membrane. There you go. And here's a really nice picture of it. You can see that the vesicle is anchored and it is releasing its secretion out of the cell. So the use of a vesicle to allow for items to exit the cell is called exocytosis. All right, I will hopefully see you soon or speak to you soon for part 2. Enjoy the rest of your day.